Shall I tell you? Shall I tell you when you're 5 minutes from 20?
Oh, yeah. Can you guys hear me? Is the mic on? All right. So thank you so much for the organizers to invite me here. Thank you for the Bulgarian Academy of Sciences and the National Science Foundation that made this possible. I kind of feel like Oumuamua that dropped into the solar system. I feel like I came close to your community. I really enjoyed it. Took 20 pages of notes sitting here, and it has been really great. But I'm not a life in the universe person. I'm a biologist. So this is our life at University of Utah. So we are somewhere in the middle of the United States. And that is roughly where that red circle is. That's where roughly my lab is. And we have a virology group, and today I'm going to be presenting to you work that is in collaboration with two of my colleagues, Professor Michael Vershinin, who does a lot of viral mechanics and looks at viruses and stretches them and does things, and Professor Shanti Dinyat, who's really an expert in high pressure, and I think that there may be some things that will be interesting for you there. Now, why am I here? Well, you know, about 2-3 years ago, there was a huge pandemic. Viruses wreaked havoc across all nations, and we started shifting and making SARS-CoV-2 viral particles. These particles have been extremely useful. We can track and understand how the envelope of SARS-CoV-2 helps deliver its genome into cells. And here I want to acknowledge the Professor Stoyanov, who has been here, and his group that we are actually in a very active collaboration to try to dissect that mechanism. But I'm not going to be telling you anything about that today. So today, I want to talk about my understanding of origin of life. And I have to make this very clear. I am not an RNA world person. I have known about RNA world, but that's not what I'm going to be talking to you guys about. I'm actually not anywhere close to the primordial life, so please do not take me for somebody who wants to profess about how viruses are the first things that were made. That's not the point here. What I want to talk about is, I want to talk about my understanding of central dogma, and then I'll introduce to you the retroviruses and specific RNA protein complexes that we have found in retroviruses that may be of your interest. I'm also going to talk about negative strand RNA viruses. These include Ebola and VSV and things like that. And then I'll tell you a little bit about a very interesting enzyme, RNA-dependent RNA polymerase, that may be of interest to your community. And then I would propose some crazy experiment based on viruses and see if there are any takers. At the end of the talk, I'll also present data on water under pressure, and I'll show you how interesting water is when you actually shock it. So you have been watching this movie. I mean, I didn't really have to have this movie on. Everybody talked about central dogma, but there is something cognitively very interesting about this movie. So when you watch it, of course, central dogma says from DNA you go to RNA, but how do you do that? You actually create these complex machines that sit down and read the DNA and make RNA. And then we have RNAs released in the nucleus. This, to me, according to my understanding, is where our brain stops functioning. We do not understand what happens to the RNA afterwards. We can't visualize things that are more than 3 components. So for us, it becomes part of a soup. It goes into a nuclear soup, and then it comes out of a nuclear soup and is fished out by the ribosomes, who assemble on it and work on it, and we understand that. We have a movie of how they do it because there are components that you can visualize. So follow me here. I'm trying to kind of make a point about our understanding of what's going on versus what's really probably going on. And it's a little bit like a 2-dimensional person trying to understand 3 dimensions. So I have 2 points that I'm going to highlight, and these are the soup points. So whenever you have a structure that is working, okay, we understand it. After that, we suddenly don't understand anything. And then again, we have an assembly that we understand, but then after that, we don't understand anything. And I want to emphasize here that that may not be actually what's happening in these systems. This is actually coming from our cognition of the system because we can't hold more than 3 things in our brain. Now, I have an example here. So, as you know, crystallography has been around for, from the time of Bragg, and we have had crystal space groups that have been very well defined. However, it took until 60s and 70s for people to start realizing quasi-crystals. So these are crystalline patterns that never actually completely repeat, but they're still capable of filling the surface. And it was actually awarded a Nobel Prize. Great observation, fantastic. Now, I want to take you to old Persian masons who were putting together masonry that was resembling, that was a quasi-crystalline pattern hundreds of years ago. And I want to ask you this question: do you think those masons actually knew what they were doing? And my short answer to that is no. They were just making patterns that were repeating themselves. And I want to say that biology is built by those masons. It's not built top-down. So maybe our focus on trying to understand biology by thermodynamics is completely ill-footed. What we really need to understand are the machines, are the out-of-equilibrium machines that have been running for a very, very long time and happen to share a cytosol. Now, how do they happen to share a cytosol? How did they hijack the membrane? Those are very, very relevant questions. But fundamentally looking at it is a different way of looking at central dogma. It does not mean RNA world. It just means you have a collection of out-of-equilibrium people, crazy machines that are running around doing things, running against each other. So what I want to introduce to you is a handle into those machines. Because these handles, these viruses, these RNA viruses, have been with us forever. Actually, I want to say this to grab your attention. So all of you who are sitting here, 5% of your chromosomal DNA are dead retroviruses. So these are retroviruses that have deposited their genome into the genome of your ancestors way before. So these things have co-evolved with us like crazy. They were first identified in electron microscopy, hence their very funny names, like a filovirus or a coronavirus, for example. And these are only the ones that make us sick. So if you think about how many viruses there are are so many different ones, but we have only characterized the ones that made us sick. And it was mainly done by EM until the revolution of molecular biology in the 50s and 60s. And people like David Baltimore got their hands onto this. And they realized that, shape is not everything. And these things are actually organized according to the genome. And, maybe a lot of you would not know, viruses do not actually need DNA genome. They can have negative strand RNA genomes, or they can have positive strand RNA genomes. They can also have a DNA genome. So in terms of understanding viruses, it's almost like understanding zoology. Okay? You can get lost very fast. But what David Baltimore did is that he broke them down into what genome they use and what enzymes do they need to process those genomes to make them compatible at living in cytosols of the eukaryotes. And so there are 2 viruses that I'm going to be focusing on. One of them is going to be HIV, which is a retrovirus. So my lab is funded on HIV research. I'm actually funded through NIH to study infectious HIV propagation. And so this is really close to my heart. And then for this conference, I'm actually bringing out some research we did on a negative strand RNA virus, because I think that may be a really cool molecular machine that this community may be interested in taking apart. So once David Baltimore did this characterization, you look at the same viruses that were previously characterized, and you realize that, for example, the Ebola virus and the rabies virus and the vesicular stomatitis virus, under their skin, they're the same virus. They're the same negative strand RNA virus. And so I'll be telling you about that replication. So the green one that I have selected here is the vesicular stomatitis virus. That's going to be what I'm going to close the talk with. And the red one is HIV, is a retrovirus. So let me bring you into HIV assembly. And I don't want to bore you with a lot of molecular detail because this can be extremely detailed and very, very overwhelming. Regardless to say, HIV has some very interesting things, and that is, once HIV assembles in an infected cell and buds out, it actually buds out and is an immature virus. And for all of you thermodynamic lovers, that basically means that it's a high state of energy. And within a couple of minutes, it would digest its internal guts and create this beautiful fuel-laden cone that encompasses this whole 2 genomic RNAs bound by specific enzymes. So the goal of the virus is to basically pack everything into these vesicles that are on the membrane and make them so that they can mature. Without that mature core, HIV is not infectious. So it's very, very critical. Most of my funding actually comes from understanding that process. And what I have at the bottom is our soup, our understanding of the cytosol. And you have ribosomes that are, can I have a laser pointer? Alright, so these are my ribosomes. That is HIV genome. HIV genomic RNA, you can think of it as a huge piece of mRNA. Think of it like that. It's really not that, but you can think of it because ribosomes walk in and they start translating from the genomic RNA. At what point there is a translation to assembly has been a question, big question, molecular question. But anyway, ribosomes are working here, they create these proteins, GAG and GAG-POL, and then they assemble into the virus and eventually release from the cell. This view of HIV assembly is informed by our understanding of central dogma. You can identify the soup. So everything goes into the soup, and then you have some complex assembly that you can kind of put your brain around and understand. Now, I want to challenge that. So I'll show you how we challenge that in the lab. So we created cells, we quantified cells, and understood how many of those genomes can we have inside a single cell that would still be assembling viruses. And then we took our understanding of HIV biology is tremendously complicated and well detailed. So we can make very specific mutations, for example, in the HIV protease, that would kill the infectivity of all the HIV genome, all the HIV virions that come out. And we created a classic competition experiment where we added this wild type genome. So this is the genome that would basically make this gray one. It's a genome that would make fully infectious HIV virions. And then we started titrating these mutant genomes. And if you're subscribing to idea of a soup, at some point your soup would get completely poisoned. So if you make proteins, if you make anything out of that soup just has 100 times more bad proteins. The beauty of this work was that we were able to push this 100-fold. So we could add 100 times more mutants than there were wild type HIV viruses. And behold to you guys, so these are infectivity plots. I'm not going to go into the details of molecular measurements, but you can basically measure the infectivity of these virions as they come out. The infectivity is plotted here. The dilutions are plotted here. And one thing that is very clear is that this plot does not go to zero. So infectivity actually traverses along a very peculiar theoretical prediction line. This theoretical line that I have here, that I have in all of my graphs, is the fraction of virions that are going to have a double wild-type genome. So somehow our infectivity just was traveling along that, not for all the proteins. The surface proteins, which are known to be assembled and trafficked through the Golgi, did not do that. Okay? So they were just dying off very, very fast. And we thought, okay, maybe this is something that just the protease does. And we just went in and we started mutating other important key elements. I'm not gonna bore you with all of that detail, and we could see that either the infectivity tracked one good genome or two good genome. This is fundamentally not agreeable with the idea of the soup in the middle. So you need to have genomes that maintain contact with the proteins that were translated from their molecular selves. And I think that this community is actually interested in that, because what you're seeing is a genome that actually maintains contact with the protein that was translated from it, so it can create some kind of a genetic feedback. Now, we modeled this thing, and these are very simple Monte Carlo modeling. This is something that your community knows exactly very well how to do. And at the end, we could actually fit all of our infectivity data and predict that the genomes of HIV were maintaining exactly the number of proteins that was essential for their own activity, and then sharing the rest. So the virus could be benevolent, it could share and rescue other progeny if they were defective, but if the virus was in an environment that was hostile, it had enough of its own proteins to get out. Now this is important for anti-drug resistant mutations, because when you have a drug, it's basically acting like a protein is mutant and you can't run away from it. The next case that I want to highlight for you, and this was kind of like a segue to get here, are negative strand RNA viruses. And these are, that's the Ebola virus, this is VSV. These guys are fascinating. One of the things that they have is that their negative strand RNA genome is fully encapsulated in protein. You can actually take this genome out of the virus and leave it on the bench. It's a piece of RNA, but it would nothing would happen to it, because they're so tightly bound with N protein that no RNAs can get in there. And the polymerases of the virus are special. These are the only RNA-dependent RNA polymerases that we know of. In your bodies, you don't have these enzymes. You don't have RNA-dependent RNA polymerases in your body. All of your polymerases are DNA-dependent RNA polymerases. So they make RNA out of DNA. Virus packages about 50 copies of this enzyme into its bullet. And it starts, and the genome is linearly organized. The polymerases walk in from the 3' end. They start making mRNA. And then they reduce the amount of mRNA creation as it will go further down. Once they have enough protein, they actually replicate the whole genome. So the whole replication of VSV happens in the cytosol, does not require any DNA. The only thing it requires is ribosomes. So we have been after these polymerases for a long time, and we kind of localize them within the virus. This is structural biology, or really not completely irrelevant, but I just want you to know that these have been really of big interest in our lab. And these polymerases, these are the polymerases that are purified, and you can see them. They are very loosely structured. So they have multiple domains. It's very difficult to get a structure from them. And one thing that we did, that after we identified the polymerases, we were very interested in their mechanism of function. We wanted to know, how do they actually do this replication in the cytosol? And the first thing we did is that we opened up the virus, we put it on top of a gradient, and spun it with tremendous amount of G forces. And what happens is that the mRNA is very heavy, and it goes way down, so it travels through a lot. But a lot of other things, like the membranes, everything floats. And what we noticed was that our mRNA, under physiological condition, was bringing down with it all of the polymerases. So if you think about it, this mRNA, 10 kilobases of RNA with all of these proteins, maintains the 50 polymerases in a grip, 1 nanomolar dissociations. Actually, that's very loosely measured. And so we had to model it. We had to think about all the possible ways that the polymerase can actually work. And these models have very clear predictions, because they have a promoter at the 3' end. The polymerase needs to find it. Polymerases walk. They can collide with each other. and then you have offerings because of collision, and so you can come up with different models that would have different parameters. And what you're looking for is basically transcription. So you want to know how much mRNA can this model make with the various conditions you have. And you also have one measurement, which is when you stop everything, the polymerases are not supposed to fall off. They're supposed to just stay on. And needless to say, I'm just going to walk you over to my favorite model. This is a non-colliding polymerase model. So the polymerases in this model can actually be transcribing and walking on the other side of the mRNA at the same time. Therefore, no polymerase is actually released from the genome when these polymerases are working, which means that this machine could be an infinite dilution machine. So you can have this machine working, and you can have a media change, and the machine will still be working as it was. Which brings me to the idea of compartmentalization. So this is a guy who you can actually take, very likely, and you can basically have it in the lab, add your polymerases or Vincent's cocktail, and then it will be able to replicate. It will be able to genetically replicate itself completely, and you would not need a membrane for it. It can just do that. Now you would ask, I had a long conversation after listening to you guys for 4 or 5 days. I actually called one of my good friends in California. He's a negative strander. And we were talking and said, why on earth would you want to do something like that? Like, this is not virologically interesting. But then we thought, what would, because VSV is highly evolved. So it's one of the highly evolved viruses. Why would you do that? And the idea would be to, well, you can in vitro evolve it, you can feed it some of the chemistry that is interesting and see if it evolves to take up that. But the more interesting thing would be to see if it de-evolves. Okay? If you passage this in the test tube and you keep doing this for a while, would the VSV lose its N protein? Would it become closer to its ancestors? Because now it doesn't actually have to compete in our cytosol with the RNases, with all the crazy enemies that it has. And remember, I'm thinking about this as a toy, and as out of equilibrium toy model. So I'm going to leave you with this thought, and if it's interesting for you guys, then please come and see me or email me. I'm very, this is an idea that has come up during the meeting, so it's partly due, I owe it partly to you guys. Now, I want to also highlight something else that we have done in the lab, just to kind of bring out water as this really crazy thing. So this is a collaboration that comes from chromatography, okay? So you have to realize the origin of this collaboration is the fact that we do a lot of chromatography in the lab, and chromatography always requires cryo, and it is not compatible with high-resolution microscopy, live or high-resolution microscopy that we do in the lab. And there was suggestions that water can create these amorphous conditions, amorphous freezing at really high pressures. And so we wanted to verify that. So we actually went in, really thinking about being able to create samples for cryotomography at room temperature and being able to freeze things within milliseconds. So if you look at, unfortunately this projector is crazy bright, but this is time in milliseconds. And you can see that our pressure cells, the one that Dr. Gimian was holding in her lap, in her hand, goes from atmospheric pressure to about 2.5 GPa. So GPa is gigapascals. Gigapascals is something around, I would say, 200,000 liters of water. Okay? So it's not center of the earth, but it's definitely 10 times more than the Maginot Straits within milliseconds. And so we can study phases of water. Now, why would be this interesting? I mean, these are impact, and you guys can think about this. I don't, my imagination is maybe less than you guys' on this. And because you have been looking at these things forever. And so you can do Raman spectroscopy, you can see the Raman spectra of the water, and can actually visualize the transparency of the water. And we can basically hit right at around, between 1.5 to 2 GPa water becomes HDA water, which is completely clear, and it has a proper Raman signature. Now, the reason why we have this paper is that there are actually crystalline pieces of I6 that are embedded in this, and it was very interesting for us, so we went to argon, we finished it up. But anyway, water by itself is a very phenomenal thing. So right now I think I'm over time a little bit, so I would like to thank the current members of the team that we have in Utah. I would like to highlight our FEI collaborators, which are building microscopes, and Jacques is our vice person I was talking to last night, and Ross Rubiak is in Argonne, our funding, NSF, and NIH, and Thermo Fisher, and the Argonne National Labs. And thank you so much for being such a good audience. And if you are interested in what we're doing, please feel free to email me and join our team. Thank you.
Thank you, Saveez. Thank you. Any questions pertaining to the talk? Herb?
I'm just curious, does the RNA virus that you described, how does it do replication of its RNA? It would be the wrong parity strand if it just
That's
used
right.
the RNA.
So it does that.
It does it twice, or does it?
Does it twice.
That's interesting.
Yes, yes, and it doesn't start doing it until it has enough N protein in the blood. So because each time it replicates the genome, the polymerase needs to shove it into the N protein. So N protein the genome is never actually naked in the cytosol. It's always encapsulated fully in a protein cleft.
Thank you, Saveez. Any more questions?
Hello, Eric Roberts, Cambridge from National Research Center. I have a simple question, maybe I just missed it. I was wondering how, in the first example you described of HIV, how contact is maintained between the genome and the protein, whether you know or if you are simply have some speculation that you're interested in hearing it, and also you think that whatever the approach is may solve the soup issue at the margin of life in a similar manner.
So thank you so much for that question. That's very insightful. That has been driving us crazy because we are, by definition of biological sciences, we are now all very fascinated by complexes and specific interactions. And so we've been looking for those. There are 2 points that I want to talk about that. Then you can have, because the RNA is about 10 kilobases and collapses into a 100 nanometer ball, you can actually have, and these proteins are bipedally. So you have 2 domains that bind RNA. So you can envision that they would be transiently walking in this RNA cloud. I mean, I just made that word up, but that would be like the RNA cloud. And so that may actually be something like that. But the more important one is that I believe that these RNA pieces that are actually embedded in these viruses are not just important for their sequence. I believe that RNA is misunderstood. RNA sequence function, sequence structure is understood really well in short hairpins and hairpin formations and things like that. These are 10 kilobases long RNA pieces that have really complex breathing modes and really complex physical things that we absolutely do not understand. And now, how do we build the technology, build the thinking to be able to understand an RNA molecule in that size and understand its breathing and protein interactions would be something that I'll be doing probably for the rest of the life.
Thank you, Saveez.
So, some people have argued in the literature, for example, there's a paper by Bader and Lascano in 2002 that the origin of life happened in some cold environment. And the argument they used was that RNA prefers cold environments because, for example, the mRNA vaccine from Pfizer and Moderna, you have to store it at cold temperatures, otherwise it decays. But if you protect it with proteins like these,
and you can see how long can it last. And this is a highly evolved RNA molecule. And I would think part of its complexity is its own ability to further evolve, because it's not the end product. So whatever piece of RNA, whatever this enigmatic folding thing is that I'm talking about, is embedded there. And so if you just de-evolve it, it will just basically relax. And there may be different temperatures we can just study. Now this is all, okay, this is science fiction. Okay, so I have to admit, I'm just, my imagination running wild, being embedded in your community for 5 days. As I said, I've taken 20 pages of notes and shared it with everybody back in the lab. So it's a little bit of going on a limb, but that's the point of these meetings, not that we just speculate a little bit.
Thank you.
Thank you, David. Any more questions? Okay, if not, let's do the following. Let's thank the previous speaker, which I think we forgot to thank him, and Saveez for wonderful talks. And if you want to learn more, please talk to them right now. I'm going to give